I'm going to talk about collagen, the different types of collagen, the synthesis, the degradation, and some diseases that are associated with it. So first, this is just the cell, I have this whole cell drawn out, and this is just the biosynthesis of collagen. We're going to talk about this after. I'm first going to talk about some general aspects, some salient traits of it, some important factors. Okay, so first, some general things. Collagen is the most abundant protein. It's the most abundant form of protein. And there's two different types of this. There's a globular protein and then there's a fibrous protein. Globular proteins are for enzymes and stuff like that. They're circular, they're round. But fibrous proteins, they're thread-like. They're collagen. So they're thread-like structures which bundle together and they form collagen. Okay, so collagen, it's composed of 3 polypeptide chains. So we have 3 chains like this. Let's see that. 1, 2, 3, right? And they're going to form together, they're going to twist up, and they're going to form a triple helix. So instead of a double helix, they're going to be like this. Triple helix, right? And I have it drawn right here. Okay, so how is this triple helix connected? They're connected via hydrogen bonds. And these hydrogen bonds are mainly how the protein is formed. It is interchain connection, so it's in the chain itself, right? From what's it called? It's from chain to chain. So 1, 2, 3. Each chain is hydrogen bonded. That's why it's called interchain. Okay? And then there's different types. We have type 1, type 2, type 3, type 4, type 8, type 9, and type 12. But I'm going to go over the types of collagen after. I just want to go through the basics of all of it first. Another thing we could talk about is some saline properties. So it's composed of 3 alpha polypeptide chains. So I said it was just 3 chains, but it's an alpha polypeptide chain. So an alpha is just like this, right? Just one straight chain. And the chain is mainly composed of glycine. 1/3 of the whole entire chain is a glycine residue. So the residue goes like this. It goes glycine, XY, glycine, XY, glycine, etc. So every third structure is going to be a glycine residue. Why is it going to be a glycine residue? So we have to remember that glycine is the smallest amino acid. Why? Because this is the structure of glycine. It's got a bond there, an NH group, right? And there's the side chain. The side chain doesn't have... there's no amino acid, it's just a hydrogen side chain. So knowing this, what do we know? It doesn't kink. Glycine is so small, it forms kinks because it's so small. Why is this important? Because this is going to aid in the triple structure. Like the triple helix, this is going to aid in the binding of the different proteins with the H on the hydrogen molecule coming off that branch is going to aid in interchain bonding. Okay, so also, the X, so we have glycine, XY. The X is usually proline, and then the Y is usually hydroxylation or hydroxyproline. We have to remember that hydroxylation and hydroxyproline are not normal amino acids, they're derivatives of amino acids. Okay, so we have translation, right? Well, we have DNA replication to transcription to translation, right? And after translation, we could do different modifications, right? This is post-translational modifications. Post-translational modifications is how we're going to form hydroxylation and hydroxyproline. Okay. So why is that important? These amino acids also aid in the triple helix structure of the collagen residue. Okay. We could say hydroxylation. This is an enzymatic, so it's used for glycosylation. And then hydroxyproline, they stabilize the triple helix structure. Okay, so the lysine is for glycosylation, and then the hydroxyproline is for stabilization of the helical structure. Okay, now for collagen synthesis. Collagen synthesis is collagen biosynthesis, technically. It's how we're going to take amino acids, right? We're going to break them, we're going to form a protein, and how this protein is going to be transformed into a collagen substance. So there's 3 different ways. We have, not 3 different ways, but 3 different types of cells. So for fibroblasts, we know we're forming collagen, so this is going to be a fibroblast cell. If we were forming bone, we would do osteoblasts. And if we were forming cartilage, we would be in chondroblasts. Okay. First, we have to remember that in the cell, proteins are transcribed where? So we're going to go through DNA replication in the nucleus. Then we're going to go through transcription in the nucleus, right? We're going to form translation. So we're not going to complete the steps of translation. We're going to send this mRNA molecule outside of the nucleus, right? When it leaves the nucleus, it could go two different ways. It could go through a signaling pathway and a non-signaling pathway. For collagen, however, we want to use the signaling pathway. Okay, so this mRNA molecule is going to have a signal sequence on it. So you have this little blue signal sequence on it. This signal sequence, right, is going to be part of step 1. So what is step 1? Okay, so we have a polypeptide precursor of collagen, right? It's in fibroblasts for connective tissues, like we said. The nucleus makes the protein. It leaves the nucleus as mRNA. Okay, we know that's how it works. Now, step 1 is the formation of a pro-alpha chain. Okay, how do we form a pro-alpha chain? So, when the mRNA leaves the nucleus, right, we're in a pre-alpha chain. It's also known as preparo-alpha chain. So in the nucleus, it's a preparo-alpha chain. However, when it leaves the nucleus, the mRNA gets a signal sequence to attach to it. It binds to it, right? That signal sequence is going to add, or it's going to put ribosomes onto the rough ER. When it puts the ribosomes on the rough ER, it's also going to put a signaling cap on the mRNA right over here. This cap is going to bring that mRNA into the rough ER, and it's going to turn it into a pro-alpha chain. So when the mRNA goes into the rough ER, that cap gets cleaved off, and when the cap gets cleaved off, we now have a pro-alpha chain. Okay, now that pro-alpha chain is going to go through hydroxylation. What is hydroxylation? We're trying to add more hydroxy groups to things. Okay, it's going to occur in the rough ER, so it's going to occur over here, and it's going to occur on the Y position. So remember what we said? You have glycine, X, Y, glycine, etc., right? So it's going to occur right on the Y position of this amino acid chain. Or this polypeptide chain is what I should say. Okay, so it's on the Y position, and the amino acids it's going to code for is lysine or proline. Okay, why lysine or proline? Because the Y position makes up hydroxylysine or hydroxyproline. So if you hydroxylate it, you're making it hydroxylysine, hydroxyproline. Okay, so in the pro-alpha chain, why? We want more hydrogen bonds, and for what? Okay, so these hydrogen bonds increase the strength of the newly formed triple helix. So we said you need interchain hydrogen bonding, right? So you have the bond like this. This is a double string. This is a triple bond, right? Really, I can't draw it too well. But if you have this triple bond, you have more H's. The more H's you have, or the more hydroxy groups on it, the more hydrogen bonding you could have, the stronger the triple helix is going to be. They're going to be bound to each other much more stronger. Okay, so that's step 2. What else does this help with? You need it, in order to do this, you need 2 different things. Okay, the enzyme that you use is called pro-lysyl or lysyl hydroxylase. So pro-lysyl or lysyl hydroxylase, no, it's prosyl, I'm sorry. Prosyl hydroxylase or lysyl hydroxylase. And this is going to do what? It's going to add the OH group. But you need an enzyme to do this. Not an enzyme like, that is the enzyme, but you need cofactors. So what do you need? You need ascorbic acid, which is also known as vitamin C. Or you need iron, and you need a molecule of oxygen. Okay, if you add vitamin C, so Vit C, iron, and oxygen, right? You're going to hydroxylate the lysine or the proline, and you're going to turn it into hydroxylysine or hydroxyproline. Now, a good way to remember this, so when it's hot outside, you want to drink what? You want to drink something that hydrates you. So you want to drink, say, lemonade to quench your thirst, right? So lemonade, so you hydroxy, right? adding like water, you could think of it as. Lemonade is what? Vitamin C for what? Lemons. So the hydroxylation is, you're thirsty, you drink lemonade, you add vitamin C or ascorbic acid, right? Now, if you don't have enough vitamin C, you have a condition called scurvy. Scurvy is a lack of vitamin C. What happens if you have a lack of vitamin C? You can't hydroxylate the lysine or the proline, so they can't be hydroxylated, and this is going to affect the rest of this process. This rest of this process is forming collagen. So if you affect the beginning processes, you're going to affect collagen synthesis, and you're going to lack collagen. Okay, next, glycosylation. This is the third step. So we did hydroxylation in the rough ER. We also did what? The signal sequence coming off of the rough ER forming the pro-alpha chain. Okay, the next step is called glycosylation. It's also going to be in the rough ER. Glycosylation, what does it do? It adds a glucose or a galactose monomer, right? Okay, and what is it added to? It adds it to hydroxylysine only. Okay, so it only adds it to hydroxylysine. Why? So hydroxyproline, if you see here, what is he going to be used for? Hydroxyproline is just adding more hydrogens to the proline molecule, or the proline amino acid. And what do we say about the proline amino acid? It helps with the structure. It helps with the hydrogen bonding because it's very small. It's not very small. It's a very, what is the word? It forms kinks because of that amino group, the 5-ring structure. So if we have more hydrogen bonds on this kinked group, it's going to help with interchain bonding. It's going to make it even stronger. Okay, so we don't want to affect him. We're going to just affect hydroxylysine. We're going to glycosylate him. Okay, after we do that, the protein molecule is going to leave the rough ER, right? And he is going to undergo like different types of synthesis. It's going to be known as assembly and secretion. So what is the assembly and secretion? It's going to add an N and C terminus. So we know in amino acids it's read 5' to 3', N to C terminus. Same thing. We have an N terminus and a C terminus, right? And it's going to be made up of disulfide bonds. Okay. So now, after that, we're going to have step 5, which is going to be extracellular cleavage of pro-collagen. What is extracellular cleavage of pro-collagen? Okay, so after the protein undergoes the N-terminal and C-terminal addition, right? This is step 4. After it undergoes step 4, it becomes pro-collagen. It is called pro-collagen. Okay, so now that pro-collagen is going to go into the Golgi apparatus, and it's going to get packaged. So he's going to be packaged in a vesicle. That's what step 5 is going to be. Just packaging the pro-collagen into a vesicle. Now, he's going to come out of the vesicle, right? After he leaves the vesicle, this is, okay, step 4 is pro-collagen being packaged into a vesicle. Step 5 now is this vesicle coming out of the cell. So I should write step 5 right here. Vesicle coming out of the Golgi apparatus, right? When he comes out of the Golgi apparatus, he undergoes certain changes. What are his changes? So he undergoes a change called NC Pro-Collagen Peptidase. So the Pro-Collagen Peptidase is the enzyme, and he's going to cleave off the N. So say you have an N terminus here and a C terminus here. Pro-Collagen N to C peptidase cleaves it off. So right here, these N and C gets taken away, and it forms a collagen monomer right here. This is called tropocollagen. Okay, so we have what? Step 1, mRNA gets a signal sequence on it, goes into the rough ER, becomes pro-alpha chain. It's a pro-alpha chain. Okay, now step 2, hydroxylation. Hydroxylation, you're adding hydroxyl groups. Step 3, glycosylation of only hydroxylysine. Step 4, the collagen that comes out after glycosylation and hydroxylation gets an N and C terminus put on it, and he gets packaged and put into the Golgi apparatus. That's step 4. Step 5, NC Pro-Collagen Peptidase comes and cleaves off the NC terminus of this structure. Now we have tropocollagen. Tropocollagen now is just placed randomly. He is randomly placed, and he is forming what? This is called the formation of fibrils. So if we have the Pro-Collagen, I don't know why I have Pro-Collagen right here. Pro-Collagen is going to form collagen fibers. He's randomly going to be placed all over the place. Okay, what do I mean by randomly placed? I mean it's quarterly staggered. So that means 3/4 of its length is overlapping someone else. So they're overlapping each other. That's how the collagen fibrils are going to form tensile strength. They're going to become very strong with this overlap. Now, that was step 6. Step 7 is going to be cross-linking. What is cross-linking? Okay, we know we have lysine and proline, right? We also have hydroxylysine, hydroxyproline. What's going to happen here? We're going to have something called lysyl oxidase, and he's going to deaminate the lysine, the hydroxylysine, or the hydroxyproline monomers. He's only going to do it to the lysine. He's not going to affect the proline ones. So he's going to take lysyl, or lysine, right? And he's going to form, right over here, allylysine. So lysine is going to become allylysine via lysyl oxidase. Now, we have a deamination one. It's going to deaminate hydroxylysine to hydroxyallicine. What is this going to do? It's going to form like little bonds, so the covalent crosslinks. So you see I have collagen fibers. We're going to have covalent crosslinks now, making the collagen fibers stick together. They're going to make them much stronger. So this lysyl oxidase deaminates the lysine and the hydroxylysine, making them form together and forming crosslinks, covalent crosslinks. Okay. Now, before I move on, I just want to explain one more thing. I just want to explain in like a flow chart. Okay, so we have step 1 is preparo alpha chains going to a pro alpha chain, right? So right over here would be the first step. Step 1, preparo alpha chain going down and forming a what? Pro alpha chain where? In the rough ER. Okay, this pro alpha chain is now going to undergo hydroxylation. This hydroxylation is going to do what? It's going to take proline and lysine using vitamin C, right? Vitamin C, what else? Iron and an oxygen molecule, right? And it's going to form hydroxyproline or hydroxylysine. We said proline goes to hydroxyproline, and then lysine goes to hydroxylysine. Now, after this, what could happen? Hydroxyproline undergoes hydrogen bonding, right? Interchain hydrogen bonding. So we have the triple helix structure. We have a stronger bond now. Also, hydroxylysine undergoes what? Glycosylation. What does this mean? It's adding a glucose or a galactose molecule onto the chain. And where is this? Both of these taking place? They're both taking place in the rough ER. Now, after that, we have Pro-Collagen chains, right? The formation of pro-collagen. What is this? This is the assembly of disulfide bonds. So remember, we have disulfide bonds on the N and C terminus. So the N and C terminus is going to take, are going to form using disulfide bonds, and we're going to form what? Pro-collagen. That's how pro-collagen is formed. Pro-collagen is then going to do what? Go into the Golgi apparatus, and it's going to be packaged into a vesicle. This packaged vesicle is then going to get what? Excitosed. So step 4 did what? Step 4 did assembly of the molecule, and it also did excretion, right? They kicked it out, secretion. Okay, that's step 4. So we have assembly of pro-collagen into Golgi apparatus, pushed out into the extracellular surface, right? Now step 5. What is step 5? Step 5 is going to be extracellular cleavage. What is that? Pro-collagen is then going to get what? The N-C terminus is going to get cleaved off using N or C. So it would be N pro-collagen peptidase or C pro-collagen peptidase. You need to use both of them. I have it right here. N or C pro-collagen peptidase cleaves off the N terminus and the C terminus, forming this right here. What is that? Tropocollagen. Okay, so now we have tropocollagen. What is tropocollagen going to do? He's going to form collagen fibrils, right? The formation of collagen fibrils. And what are those guys? These guys are right here. This is the overlap of the collagen. 3/4 structure, right? That means they're quarterly staggered. That means 3/4 of them are overlapping each other. You should probably see it like this. It would make it a little more clear. A little bit longer. Okay. Now we have step 7. What is step 7? It's the cross-linking formation. What is cross-linking formation? Linking the collagen fibrils to each other. How are we going to do this? Okay. So we know lysine is going to turn into allylysine, right? Using lysyl oxidase, which needs copper. This is very important. Remember that lysyl oxidase and oxidative deamination, they need copper. This is a copper enzyme. Okay, when that happens, hydroxylysine becomes hydroxyalysine via oxidative deamination. And what are they going to do? They're going to cross-link. So lysine is going to bind to other allysine molecules, and hydroxylysine are going to bind to other hydroxyalysine molecules. What is this going to form? The mature collagen. We said it right here. These covalent crosslinks forms mature collagen. So these mature collagen fibers is what will be used by the body. Okay, so now I'm going to talk about the different types of collagen. So we have a couple different types. We said it before, we have type 1, type 2, type 3, type 4, type 8, type 9, and type 12. So let's break it down first. There's the first 3 collagens, type 1, type 2, and type 3, are all fibril-forming collagen. What is fibril-forming collagen? This is the collagen that uses fibrils. So it needs collagen fibrils, which we talked about, that whole entire process with the cross-linking. That's the main 3, 1, 2, and 3. So now the main one out of the main 3 is type 1. This is the most abundant form of collagen. So he's the strongest, most abundant, and he's for tensile strength. Where is he? How is he created? So he uses 2 alpha 1 strands and 1 alpha 2 strand. I don't know how important that is, but that's something. What does it make? It makes his bone, tendon, cornea, and skin. Okay, how can we remember this? This is going to sound stupid. I found it from a YouTube video, but it helped. Bone, number 1 is in the name. Tendon, number 1, you can make. Cornea, you could form 1 for cornea. And then skin tone, 1, you could put 1 in the name, right? So number 1 is in the name of type 1 collagen. Okay. Type 2 collagen, what is it? Spongy, right? So it absorbs shock, and it's in the virtuous body and nucleus pulposus. How can we remember this guy? Another dumb one. Virtuous, and then nucleus pulposus. Also, we have to know it's made of 3 alpha 1 chains. Okay. Type 3, it's webby, and this is with pulling forces. So remember, whenever you're dealing with a pulling force, it's probably going to come from type 3 collagen. Tissues with pulling factors are what? Blood vessels, right? You're pulling blood. Uterine tissues, you're pulling urine. Granulation tissues and muscles, right? Also fetal tissues. The mnemonic or acronym that I learned to name this one is UT3, so uterine, type 3 collagen. FT3, fetal tissues, type 3 collagen. GT3, granulation tissues, G3 collagen. And BV3, blood vessels, type 3 collagen. They kind of rhyme. BV3, UT3, FT3, GT3, etc. Now, there's 2 diseases that come in the fibril-forming collagen. The first type is Osteogenesis Imperfecta. What is Osteogenesis Imperfecta? Okay, it comes from a lack of type 1 collagen. And how do we get a lack of type 1 collagen? So a lack of type 1 collagen could come from glycine, right? Remember how we have glycine, X, Y, glycine, right? So you can have a replacement from a bulky amino acid. So instead of a glycine, right, you just have a mutation with tryptophan, right? So you have tryptophan instead. You can't form this triple helix. If you can't form the triple helix, you're not going to create type 1 collagen. If you can't create the type 1 collagen, what are you going to have? You're going to have multiple defects known as fracture, like your bones are going to be weak. So it's going to be non-traumatic fractures. That's one of the disorders you're going to have. And a non-traumatic fracture is like random breaks in your bones. Not from a traumatic injury. So I don't have to be hitting the arm to fracture. I could just fracture, my body might just fracture because I have brittle bones, also known as glass bones, right? Okay. What else? We could have blue sclera. So this is a translucency of connective tissue in the eye. So you could see if you look at someone's eye right over here, their sclera, instead of being white, which it's supposed to be, it's going to be a blue color. The last one's going to be hearing loss. Why? Because if you can't form a bone, tendon, cornea, like etc. in your skin, but if you can't form bone, first off, you have bones in your middle ear. If you can't form those bones in your middle ear, you're not going to be able to hear. Okay. Our next disorder we can talk about is from type 3. This is Ehlers-Danlos Syndrome. What is this syndrome? It's from Pro-Collagen, right? The copper is affected, right? If copper is inhibited, what occurs? Well, we said copper is used for what? Hydroxyproline. And which other one? Lysyl oxidase. Lysyl oxidase and oxidative deamination forms what? Hydroxylysine into hydroxyalysine, or lysine into allylysine. Remember we said, let me see where I could put this. I'm going to put it right up here in this little box for you guys. We had lysine going to allylysine, and we had hydroxylysine going to what? Hydroxyallylysine. Okay, and they formed what? The covalent crosslinks, which led to a strong collagen fiber, right? The mature collagen fibers are formed. If copper is like not here, what could occur? Never mind. This is not even close. Wow, that's bad. Scratch that. So where were we? Blue sclera, right, is translucency of connective tissue. Why? Your eyes are supposed to be white, but instead they're blue now. Also, we said the bones in the middle ear, we had hearing loss. Okay, another type of disorder we can talk about is Ehlers-Danlos Syndrome. What is that? Ehlers-Danlos Syndrome. Okay, it's a deficiency in Pro-Collagen. What is Pro-Collagen? Okay, remember we spoke about how you go hydroxylation to glycosylation, and then glycosylation goes to Pro-Collagen when the NC terminus gets added to the ends of the collagen that was glycosylation and hydroxylated. Okay, so when that happens, if you have a deficiency in the Pro-Collagen, what can't you form? You really can't form type 3 fibers, right? Type 3 collagen is the big defect, like we have, type 3. What type of structures are these? Okay, we had blood vessels, uterine vessels, fetal tissues, granulation tissues, and muscles, right? Okay, so some disorders we're going to have is berry-aneurysm. So we're going to have like an aneurysm in our brain. We're going to have bleeding and bruising. So you're going to bleed and bruise easily because you don't have the pulling factors to aid in inflammation and repair. We're going to have organ rupture because this is also a pulling force. That's what our organs are. And we're also going to have mitral valve prolapse, right? Heart conditions. Okay, so we're going to have a lot of ruptured arteries and veins in this condition. And what enzyme is mainly defective from Pro-Collagen peptide? From Ehlers-Danlos Syndrome? It's going to be Pro-Collagen Peptidase, and it's going to be lysyl oxidase. Those are going to be the main enzymes that are defective. Also, Ehlers-Danlos Syndrome can take place in the nucleus where like mRNA is translated, but it's mainly coming from Pro-Collagen Peptidase. You're not forming what? You're not taking off the NC terminus, right? You're not using the N or C Pro-Collagen Peptidase right here. Let me show it to you guys. And collagen peptidase, right? We're not taking off the NC terminus. We can't form anything else. We're stuck at this stage. When we're stuck at this stage, we get Ehlers-Danlos Syndrome. Okay. Now, if I could put it back up. Okay. So now, another type of collagen we could talk about. Type 4 and type 8. This is network-forming collagen. So it's forming networks, right? So type 4 is in the basement membrane, right, and provides stabilization. What is this for? your kidney, your ear, your eyes, and your skin. They're more like the outer factors. It's not the ear, the hearing part, and it's not the eye, the cornea part. It's just outer factors, the basement membrane, right? Type 8 is for corneal or vascular epithelium. Okay, and this is network forming, so it's networks. The last type I want to talk about is type 9 and type 12. They're not only cartilage, they're fibrils that are associated with the triple helix and other domains. So they have more than just cartilage with them. And what do they form? Type 9 is going to form cartilage. And collagen before. I meant collagen up here. It's going to have other collagen with it. It's a triple helix with other domains of collagen. Okay. Type 9 is going to form cartilage. And type 12 is going to form tendons, ligaments, and some other tissues. Okay. Another disease I want to talk about is scurvy. We mentioned it a little bit before, right? Scurvy, I mentioned him over here. And what was he? He was a lack of vitamin C, right? So if you have a lack of vitamin C, you can't form the triple helix. Wow. Okay, why? If you don't have vitamin C, you can't have this enzyme, the hydroxylation enzyme. That hydroxylation enzyme won't make mature collagen come out. What are some disorders that is going to be involved with him? You're going to have bleeding in the gums. You're going to have loose teeth. You're going to have pedicne, ecchymosis, poor wound healing, and poor wound development. Okay, where is this common? This is common in pirates, right? Pirates are usually or used to get scurvy, right? Okay, how can we remember this? Okay, pirates are out in the ocean, right? If they're out in the ocean, they do what? We could say hydroxylation, ocean hydroxylation, right? But if they're stuck at sea, do they have fresh fruits or vegetables? No. If they don't have fresh fruits or vegetables or citrus fruits, they don't have vitamin C. Why? They're stuck at sea, so they're deficient in vitamin C. Okay, if they don't have vitamin C, we can't do hydroxylation. If we can't do hydroxylation, we can't form what? Pro-Collagen. And that's going to be scurvy. Okay. The last one I want to talk about is Menkes, right? Menkes is a copper deficiency. So you have low copper and you have low serum copper, like a copper serum. Plasma copper serum, I'm sorry. This is depigmented hair, artery ruptures, cerebral degeneration, osteoporosis, and anemia. So where would you find this? You would find this down here. This step, right? Menkes. Down over here. Lysine oxidase, right? And then the oxidative deamination, it uses copper. If you can't do, the oxidative deamination or use lysyl oxidase on lysine or hydroxylysine, you're not going to be able to form al-lysine or hydroxy-al-lysine, right? So you're not going to have any cross-linking. So this would be inhibited. If this is inhibited, you can't form mature collagen. If you can't form mature collagen, you're not going to be able to have the certain collagen functions that I just mentioned. You're going to have depigmented hair. You're going to have artery ruptures. You're going to have cerebral degeneration. And you're going to have osteoporosis or anemia.